2023 at the ALSP Community Conference in his area at UC Irvine in California. He took us on this fantastic tour of the lab facility there. Yeah, don't even know if you remember this part, but we're going through the tour. We're going through some like back hallway, going from one place to the other, and just equipment and tables and stuff just sort of scattered everywhere. Well, someone asked a question, and Rob immediately finds a whiteboard there in this back hallway, finds a marker, and goes up and just gives this incredible explanation and answer. And that's just kind of how he's always been, how I've always known him. Any of the presentations that he gives, he's just incredible at explaining things. I've seen him multiple times since then. He's able to explain complex things in a very clear way, but without losing any of that meaningful content. So hopefully you'll enjoy the presentation. Hopefully you'll get a chance to talk with him when he's not up on the podium. He's got a good person to talk to with a great sense of humor. So, Dr.
Thank you so much. My name's Rob. I'm not going to stand behind podiums. I don't like podiums. Can you hear my voice? Okay, good. So,
Zoom cannot hear without the mic.
Zoom, okay. Fine. I'll stand at the podium. All right, so when Erin asked me to come and participate in the meeting, she asked me to cover two topics. So unfortunately for you, you're going to see me twice today. The first time is I'm going to talk broadly about microglia cells and what they do. The big point that I want you to leave from this presentation about is that microglia disorders are more common than anyone thinks. And the things that this patient community learns from the research, the clinical trials, the interactions between patients, patient advocacy groups, and caregivers are going to be exemplar for many other diseases and disease patient advocacy groups, because you guys are setting the stage for what is going to become a very common way to think about brain diseases broadly, which is that the immune cells are not doing what they should be doing inside the brain, okay? So my function, my day job, is I'm the co-founder and CEO of a company called Savannah Biotherapeutics. The vision of Savannah is to reprogram the brain's immune system focused on microglia cells, to treat the diseases through the immune system lens. I'm going to talk a little bit about microglia cells that I'm really excited because my colleague, Dr. Jean-Paul Chedaravian, who set a lot of the scientific foundation for Savannah in the research lab of Savannah's co-founder, Matt Blurton-Jones, is going to tell you guys about the work that he's been doing to explore the therapeutic potential of induced pluripotent stem cell transplant in ALSP animal models. And then I'm going to come back, and then I'm going to talk to you about what the company thinks are some of the grand challenges to take this information that we're learning from the research work, from our knowledge of microglia cells, and make scalable medicines that can treat these diseases. And ALSP, I think, is something really for us as a community to pause about, because we all understand and appreciate that it is significantly underdiagnosed. This is a very big problem. To be able to bring a therapy to you folks at scale to address the real problem is a monumental achievement, period, okay? So that's the second thing I'm going to talk about, but let me start by talking about microglia cells a little bit. And I'm fortunate because Dr. Kufal gave a great introduction about where microglia cells come from, how they get into your central nervous system, what are the implications of understanding them. So I just want us to pause on that point to understand what they do, okay? All right, so as a starting point, sorry, some of the colors are not coming through as well as I should have. I tried to get them to adjust the lighting, but I'll do a lot of interpretive dancing for anything that's not represented there. I think everybody can understand that we have a peripheral immune system. These are our B cells, our T cells, our natural killer cells. These are the things that do a very good job of fighting off things like infections, or if you get a cut on your leg, those cells are very good at mobilizing and having an immune response to that. Our brain and our central nervous system has a different immune system, okay? In the ears, the eyes, the brain, and the spine, there's a different cell whose job it is to survey, make sure everything's working right, clean up any of the debris, take care of any problems. That is a microglia cell. So the microglia cell is a central conduit to immune functionality and immune preservation protection. The job of these cells is to survey, surveil, and protect your brain from any of these kinds of problems, okay? So for example, in ALSP, we all appreciate that there's a depletion of healthy microglia cells from the brain compartment. Patients are actually losing the number of those microglia cells, so you're actually losing the number of functional immune cells that are in your central nervous system. Okay, we'll talk a little bit about that, and John Paul will cover some of the thoughts around that as well. As Dr. Kufal mentioned a few minutes ago, the brain is, I think, the best example of the most complicated organ system in your body. It has the most number of diverse cells that perform many different types of functions. There's a lot of work that goes on to control electrical activity, the movement of molecules, of course, the formation of memory, the way that we see things. We even have these things called grid cells in our brain that tell us how we're moving through space and that there's a chair there. There's a lot of really complicated functionality that's happening. And the microglia cells are there to make sure that all the structural connections and cellular connections are maintained in a way that works right every time, all the time, okay? So, how many of you guys like college football? So I'm a big fan of college football. One of my favorite coaches is Nick Saban. So he says, in order for a team to work perfectly, it has to do the right thing at the right time, all the time. Okay? So immune cells in the brain, microglia, they have to do the right thing at the right time, all the time. And I think we should all appreciate how much that's actually a miracle for brain cells, and especially for microglia, because they are self-renewing cell types. That means that they will divide in your brain and self-renew their populations. Every 4 years or so, the total population of microglia that's in your brain and in your central nervous system will, sorry, I'm being told to go closer to the microphone. I'm gonna do this, okay? All right. So every 4 years, that population of cells will turn over and replenish itself. It's self-renewing. So that means it has to divide and maintain its good measure every time. It is literally a miracle for that to happen for the lifetime of someone's life, over 70 or 80 years of time, okay? So these cells, these microglia cells, these unique immune cells, they're performing very specific functions over quite a significant amount of time. So some of the basic properties, I know as clinicians and patient advocacy groups and patients, everybody here has a different set of understanding of what microglia do, depending on what you focus on. But for, I think, the sake of patients and patient advocacy groups and caregivers, these are kind of the really important functions that I think I want to impress upon you guys. Of course, they live in the brain. Microglia cells are not outside of the central nervous system compartment. There are specific protein molecules called IL-34 and CSF1 ligand that bind to CSF1 receptor. You guys are familiar with that gene, CSF1R. If those two things aren't talking to each other, then microglia cells can't survive, which is why in ALSP, if there are mutations in those genes, that gene is not working correctly, and those cells are in trouble. Outside of the central nervous system, we don't have the right components to keep microglia cells alive necessarily, so they don't survive outside of that compartment. They sense problems. They're very good at noticing when things are not quite right or not going well. They respond quickly. Savannah's scientific team published this paper, which is led by Dr. Blurton-Jones's lab a few years ago, showing that you can image microglia cells in real time inside of a fully intact brain and show that they respond within minutes to noticing there's a damage. So they will actually migrate to where they're supposed to go. They're just remarkably responsive cell types. They have this characteristic called chemotaxis. They can actually move great distances to get to where they need to be. They're highly motile in that way. They clean up damage. They're a phagocytic cell type. They're a macrophage. They're very good at getting rid of bad debris. You could think of them as very mobile garbage trucks, right? Taking things and moving them out of the way and getting rid of them. And they help to maintain brain circuits. The way they do that, one particular thing we'll focus on is that they're very good at maintaining and cleaning up the structure of neurons and axons and when neurons make synapses. So a hallmark of ALSP is that they're axonal spheroids, these clumping ups of axons that damage them. And under normal circumstances, microglia actually clean them up and maintain them. So it makes sense that as microglia cells go down, those problems arise, and it's because the immune system is not quite in the right balance. So it's something to consider there, okay? The other thing I'll mention is that they will actually do a lot of different functions depending on how they interact with other cells. I like to think of them as the Swiss Army knife of the brain, okay? They do a lot. Are you laughing at me, Aaron?
It's brilliant, I love it.
Okay, don't laugh at me.
I'm not laughing, I'm like, we need to write that.
Okay, so they're like the Swiss Army knife of the brain. So depending on what they're supposed to do or which cell type they're supposed to interact with, they can do any type of function. So they regulate the function of neurons. They support oligodendrocyte function and structure, which is why ALSP is a white matter disease. So oligodendrocytes supply the brain with myelin basic protein and have that beautiful white connective tissue that is in white matter. And if the microglia are having trouble, they have a hard time maintaining that white matter integrity. So it can be compromised, hence why there's a lot of white matter dysfunction or MRI flare dysfunction, T2 MRI flare abnormalities in white matter. Microglia are not helping to maintain that correctly, okay? They also stabilize pericyte, which are a cell type that will line the cerebral vascular structure inside the brain. They're responsible for maintaining the integrity of the blood-brain barrier and the pericyte interaction with cerebral vascular cells. They can recruit peripheral immune cells. They can clear dead cells or debris. They will monitor epididymal cells and protect epithelial cells, which protect the blood-brain barrier. And they can coordinate with astrocytes, which I haven't seen Dr. Shadaravian's slides yet, but I think he'll talk a little bit about things like gliosis or stress response. These cells are very good at maintaining astrocyte cell maintenance and stability, so they do that quite well. Swiss Army knife. If there's anything you leave the ULF meeting with, Swiss Army knife. Okay, good. I made my impact. So because they do so many things, they are the perfect cell type to just cause a bunch of problems, all right? They're a central conduit to brain health broadly. They are really critical for maintaining all of the right integrity, structure, signaling integrity, things like that. So you can imagine that if that central conduit has trouble, that there's a lot of things that trickle down from that. So let's talk about that, okay? They can influence almost every part of the brain. They are, as I mentioned, present in the ears, the eyes, the spine, and in the brain. They're also everywhere in the brain. They are enriched in white matter regions, but they're everywhere in the brain, okay? They can damage healthy brain cells when they malfunction. So as Dr. Kufal mentioned, it's really good to have a neuroinflammatory response, but if you have a neuroinflammatory response where microglia are sending out these signals for long periods of time, that can actually cause an issue over those long periods of time. So it's good that they are acutely good at responding. They can respond quick when they see something they don't like, but if they do that for long periods of time, it can cascade into causing some problems. They can help shape brain connections for better or for worse. So if they are not integral at helping neurons talk to each other, form synapses, form physical connections that then turn into the correct electrical connections, and that can malfunction, that of course can cause a problem with regular brain circuitry, and that can result in changes in behavioral and functional abnormalities that we would see in patients. And they can stay dysfunctional for a very long time. Many brain diseases are linked to faulty microglia. They're the linchpin. As I often say, as microglia health goes, the brain health goes too, okay? So those things are directly related. Unhealthy microglia genetics can drive many different diseases. Now, we focus on ALSP CSF1R because, you know, it's a very critical and what I describe as a linear microgliopathy. That is, it's a primary microgliopathy. There's a mutation in a gene that's critical for maintaining microglia health. When that genetics goes wrong, then there's a series of cascades that result from that. So it's very linear to describe it that way. But it turns out there's a lot of different diseases that are also caused by faulty microglia function, which is why I mentioned at the beginning that it's exemplar, the type of research, patient advocacy group awareness, biomarker and clinical trial design focused on microglia that is acutely centered around ALSP that I think will have such a ripple effect on the field as we start to better understand what microglia do, what they do when they go wrong, and how we think about designing clinical programs around them, okay? So for example, I won't belabor this, but a lot of the genes that are used by clinicians to make a diagnosis for Alzheimer's disease, for example, are actually genes that are enriched or exclusively expressed in microglia. So there's a focus on microglia cells that clinicians are starting to work more and more on. And even from one cell type, many problems can occur. So in Alzheimer's disease, ALS, Parkinson's disease, or dementia with Lewy bodies, mutations in microglia can change their ability to clear amyloid or clear debris. It can cause overreactive microglia to release harmful inflammatory signals, and it can even impair support for neuron health and protein handling or protein processing, okay? Now, it turns out that because in ALSP you're losing those cells, so it's a loss of those microglia, a lot of these issues are likely to be more problematic in ALSP because the cell type is actually, you have this beautiful network of cells that's in the brain and you're taking one of them away. So it becomes much more problematic. You can't see this very well, but there's a line here. And so I think getting the balance tipped to healthy microglia can actually be very powerful. So let me tell you about a couple of examples of what I mean by that. So it turns out that faulty genetics can drive microglia diseases. But it has also been demonstrated in the clinic that microglia that have gain of function protective genetics, that those microglia cells can stabilize the brain environment to never lose cognitive function, even though you have very strong symptoms of the disease and the pathology. So the hallmark case of this is there have now been a couple of cases where someone had a mutation in a gene called presenilin-1, which is 100% penetrant in Alzheimer's disease. If that same person had a mutation in ApoE that was able to compensate for that other presenilin mutation, those patients can have very aggressive pathological dysfunction, amyloid plaques, et cetera, but they never lose cognitive ability. So the microglia cells, which are the immune cells, are supercharged to protect the brain, okay? So what this means is that the tipping point is really critical here. So if you have microglia dysfunction and you can compensate for that with healthy microglia, you can actually do remarkable things on controlling brain and cognitive health. The other thing that's quite remarkable is that in a study where they were looking at superagers, so these are folks that are over 80 years old, and they had really tremendous cognitive stability as they got to be older than 80 years old. Those patients had an enrichment for protective microglia or anti-inflammatory microglia, further suggesting that how the immune cells behave in the brain is really critical for controlling what's going on with cognitive disability or behavioral disability, et cetera. Okay? So, some key takeaways that I want you to take from this. As I mentioned, I'm super excited for my colleague to come up here and tell you guys a little bit more about the basic research that he's been doing in ALSP. We're very thankful for that. So, but from here, I think it's a reasonably good introduction into what microglia will do. The brain has its own immune system, and microglia are at its core. Microglia are a central hub touching every part of the brain. They're everywhere. These things are everywhere, okay? When they malfunction, microglia are built to do lasting damage. So really controlling that is critical, and all the things that we're going to learn about ALSP in the future going forward are going to be critical to inform us. One cell type, many diseases. So one cell type can really control how things cascade, either directly or indirectly. Directly would be for primary microgliopathies, or microglia genetics is known to directly contribute to the disease. Indirectly could be that the brain is in a certain pathological state where the microglia cells are overreactive and they're over-responding in a pro-inflammatory state, and that can have long-lasting effects on what happens. And then restoring healthy microglia is powerful, and human genetics can prove it. We have not a lot of cases, right, but a good amount to just start thinking about the potential of the impact of controlling microglia genetics and how that can be beneficial. As Dr. Kufal mentioned, there's a lot of excitement around microglia replacement, hematopoietic stem cell transplantation, or other sources of microglia cells to reset the cell population, which of course would benefit the genetics. If you have a series of cells that have faulty genetics, you want to introduce cells that have healthy genetics that may be able to compensate for that. Or in the case of ALSP, introduce a group of healthy cells that can compensate for a reduction of microglia cells. So those are some of the things that we're really excited about. I will pause to take any questions. I'm happy to do so.
We're also taking questions over Zoom, so if you're on Zoom, you can type them in the chat box.
That's okay too.
You explained everything. There are no questions.
I doubt it.